Every bite of bread you eat begins a complex biochemical journey inside your body, turning food into energy at the molecular level. Carbohydrates are one of the primary macronutrients required by the human body for energy production. They are composed mainly of carbon, hydrogen, and oxygen atoms, arranged in different structural forms. From a biochemical perspective, carbohydrates serve as the most immediate source of glucose for cellular metabolism. The digestion of carbohydrates is a multi-step process that begins in the mouth and ends at the cellular level. Dietary carbohydrates are broadly classified into monosaccharides, disaccharides, and polysaccharides. Monosaccharides, such as glucose, fructose, and galactose, are the simplest forms and do not require digestion. Disaccharides like sucrose, lactose, and maltose must be broken down into monosaccharides before absorption. Polysaccharides, including starch and glycogen, require extensive enzymatic digestion. The process of carbohydrate digestion begins in the oral cavity with mechanical and chemical actions. Chewing increases the surface area of food particles, making enzymatic action more effective. Saliva contains the enzyme salivary amylase, also known as TI. This enzyme initiates the hydrolysis of starch into shorter polysaccharides and maltose units. Salivary amylase specifically breaks alpha-14 glycosidic bonds present in starch molecules. It does not act on alpha-16 bonds, which are found at branch points in amylase. The activity of salivary amylase is optimal at a neutral pH. However, its action is temporary because it becomes inactivated once the food reaches the acidic environment of the stomach. After swallowing, the food bolus enters the stomach where carbohydrate digestion temporarily pauses. The highly acidic gastric juice denatures salivary amylase. No new carbohydrate-digesting enzymes are secreted in the stomach. As a result, carbohydrates remain largely unchanged during their stay in the gastric lumen. The major phase of carbohydrate digestion occurs in the small intestine. The pancreas secretes pancreatic amylase into the duodenum. This enzyme continues the breakdown of remaining starch molecules into maltose, maltotriose, and pancreatic amylase is highly efficient and functions optimally at a slightly alkaline pH. Pancreatic amylase still cannot break ALPHA16 glycosidic bonds. As a result, branched oligosaccharides, known as limit dextrins, are formed. These partially digested carbohydrates require the action of brush border enzymes. The final digestion takes place on the surface of intestinal epithelial cells. Brush border enzymes are embedded in the microvilli of the small intestine. These enzymes include maltase, sucrase, lactase, and isomaltase. Each enzyme is specific for a particular type of glycosidic bond. Their combined action ensures complete conversion of disaccharides and oligosaccharides into maltase hydrolyzes maltose into two glucose molecules. Sucrase breaks sucrose into glucose and fructose. Lactase splits lactose into glucose and galactose. Isomaltase acts on alpha-16 bonds present in limit dextrins. Once carbohydrates are reduced to monosaccharides, absorption can take place. Glucose and galactose are absorbed through active transport using the sodium-glucose cotransporter 1. This process requires energy indirectly from the sodium gradient maintained by ATP. Fructose, on the other hand, is absorbed by facilitated diffusion using GLUT5 transporters. After absorption, monosaccharides enter the portal circulation. They are transported directly to the liver via the hepatic portal vein. The liver plays a central role in carbohydrate metabolism. It regulates blood glucose levels by storing or releasing glucose as needed. Inside the liver, glucose can be converted into glycogen through a process called glycogenesis. When blood glucose levels fall, glycogen is broken down by glycogen. Excess glucose may also be converted into fatty acids and stored as triglycerides. These metabolic pathways are tightly regulated by hormones. Insulin and glucagon are the two primary hormones controlling carbohydrate metabolism. Insulin promotes glucose uptake and storage after a meal. Glucagon stimulates glucose release during fasting states. The balance between these hormones maintains glucose homeostasis. At the cellular level, absorbed glucose undergoes glycolysis. Glycolysis is a biochemical pathway that converts glucose into pure. This process generates ATP, which is the energy currency of the cell. It also produces reducing equivalents in the form of NADH. If oxygen is available, pyruvate enters the mitochondria for further oxidation. It is converted into acetyl-CoA and enters the citric acid cycle. This leads to the production of more ATP through oxidative phosphorylation. In the absence of oxygen, pyruvate is converted into lactate. Not all carbohydrates are digested in the small intestine. Dietary fiber resists enzymatic digestion and reaches the large intestine. Here, it is fermented by gut microbiota into short-chain fatty acids. These fatty acids provide additional health benefits and support gut health. In summary, carbohydrate digestion is a highly coordinated biochemical process. It involves multiple enzymes, organs, and regulatory mechanisms. Efficient digestion ensures a continuous supply of glucose for metabolic needs. Any disruption in this process can lead to digestive or metabolic disorders.